Let's see. Hello. Okay. Greetings, students. This video is to be watched in conjunction with the live session video that was recorded for you to read about or learn about all of the class requirements. This one is on the lab report instructions and the rubric. In other words, what is expected on the lab reports. All right, since I showed my ugly mug all through that introductory video, I'm not going to show it here. So you can kind of just ignore the fact that I'm here other than my voice. All right. So let me go ahead and share my screen. Okay. So we're sharing screen. Um, I'm going to go into the classroom here for a second to show you something. All right. I mentioned in the last video that when you open up the lab report for that week, you're going to see a couple of things. You're going to see the lab report instructions, which I'm going to go over right now. And you're also going to see the lab report template. And I opened that in a new tab so that we can talk about that. Also below here is the rubric that I'm going to discuss today. Okay. So let's go ahead and open up the lab report instructions. Whoops, that's not the one I want. Oh, there we go. Lab report instructions. All right. And you can find those, as I mentioned, right there in the lab report. The lab report instructions are given to you each week. So that if you need a refresher to look at this, you can just open that up and look at it. I'm going to go over this in detail, though, and give you all of the expectations and exactly what your lab report should look like when it's finished. And if you follow what I'm talking about here, then you're going to do just fine. All right. So first of all, the lab report needs to be written in APA format, which in this case means you're going to double space the lab report. That's the important thing. Each lab report is going to go, when you turn it in, it's going to go through Turnitin, which is a plagiarism platform that we pass all our assignments through to get matches to make sure students are not copying and pasting other people's lab reports or something. Once Turnitin is done with it, a lot of times this the font and the font size are kind of lost. So I'm not really paying much attention to that. I am looking for double spacing. I am looking for APA formatting on the references. I'm not looking for this length. Most students' lab reports are much, much longer than 2 or 3 pages. So don't worry about how long your lab report is. Worry about if your lab report includes everything that it needs to include. That's what I'm looking at. I don't really care how long or short it is, as long as everything that needs to be there is there. All right. So APA format. Now, there are 6 sections in the lab report, and I do not want to see more than 6 sections, please. Do not add your own headings and your own categories and things. Just use these sections that are outlined in the instructions. That makes it easy for me and you. It makes it easy for you because you know what's expected in each section, so you can just put it in there and be pretty happy with that, knowing that you did what you're supposed to. And for me, it makes it easy to grade because I don't have to be looking around for things that are supposed to be included in this section or that section. They should be right there. All right, so your lab report needs to start out with a title page, a totally different title page, right? A separate title page, which should be in sort of APA format. I'm not real picky about this. As long as the title is centered and it's down the page a little bit like APA requires, that's good. I'm fine with that. All right. So write your title page out just like you would if you were writing a paper for another course. Then at the bottom of the title page, or when you're done with the title, do a page break. That way, the 4 written categories or sections are going to be started on a new page. All right, so page break, and then the first heading, which you must write out, you must write heading, or don't write heading, but write introduction, okay, as your heading. And then you're going to write the stuff that's included in the introduction under that. All right, so first heading is introduction. And then what's expected here? Well, what you're going to do here is you're going to include a summary of the background information, any research that you might do in to kind of figure out what you're doing in the lab, include that. Some students have come up with some pretty interesting research that I actually learned a couple things from a few students over the years. Okay, but let me go over the background on Lab 3. All right, where's that? I've got it open. There it is. Okay, so if you open up this thing here, this lab report template, you can download it. Obviously, you need to do that because you're probably going to want to use the template somewhat. You're going to copy and paste some tables and things from this into your lab report. So you might want to download it, but I'm just going to look at it so I can talk about it here. All right, so in Lab 3, notice the title, please, everybody. The title is Limiting Reagents, which could be written as Limiting Reactants. I've had students in the past with this lab who go through the entire lab never mentioning limiting reagents or limiting reactants. But notice that the title tells you what the lab is about, right? So you should be discussing limiting reactants in this lab. That's an important concept because it's the title of the lab, even though it's not really explicitly laid out here in the background. All right, so what should you include in your introduction? First of all, you're going to include any lab scenario there is. For example, there's a couple of labs you're doing where you as a scientist are doing some particular things. Like in lab 1, you messed up and you left some bottles near an incinerator and the labels came off. So you've got to identify the substances. That would be your lab scenario. And several of the labs have these. So make sure you include that because that's kind of giving you the goal of the lab or why you're doing the lab. So that should be there. Then, if it discusses certain concepts, for example, if you're discussing density, you probably want to talk about density. If you're talking about a reaction, then you want to include the reaction. So, for example, in this lab, you're actually doing 3 iterations of this reaction. So you might want to research that a little bit. So actually, you're not doing 3 here. You're doing just this one. Okay, never mind. So you want to include this reaction because that's the reaction that that's under study, and then include anything that it talks about here in your introduction as well. The lab objectives should be included. Each lab background does have the lab objectives for that particular lab included. So make sure that you write those out. If there is a formula, for example, for calculating some stuff, you want to include that formula. And then anything else in the background that you think is important, include it. All right. This is just going to give the reader of your lab report an idea of what the lab is going to be about and what you're going to be doing, that kind of thing, what the goals are of the lab, that kind of thing. All right. So let me go back to our, there we go. Maybe, come on, open up. There we go. All right, so that is your introduction. And then you have one more thing that you're going to include in the introduction, and that is your hypothesis. I think most of you have probably heard that term before, but let me go kind of through it to explain what it means. A hypothesis is an educated guess. It's not just a guess you pull out of thin air. It's an educated guess. And how are you going to find the educated guess in your lab reports? What you're going to do is you're going to read the entire lab report. And if you do that, it's going to kind of appear to you what the goal of the lab is, right? For example, if you're trying to find the identity of some unknowns, that's the goal of the lab. All right, so your hypothesis should include something about that goal. And it should, so that's what I mean about an educated guess. You're using some information to make your educated guess. You're not just pulling things out of, thin air to make a prediction. All right, so let me give you a good and a bad hypothesis. Let's say that you are identifying some substances using density, all right, like you do in lab 1. A good hypothesis would be, if I find the densities of my unknowns and compare their densities to the known values, I can identify my unknowns. All right. That states the goal of the lab. It tells basically how you're going to achieve that goal, comparing densities. That is an excellent hypothesis, and you would get full credit for that. Okay. What is a bad hypothesis? A bad hypothesis would be something like, I predict that unknown A is zinc and unknown B is aluminum. Why is that bad? Well, because you're pulling that out of thin air. You have there's no basis for that prediction at all. In other words, you would have to do the lab to come up with those answers. And you are not going to do the lab until after you've made your hypothesis. The hypothesis needs to be made before you do the lab. So that would be a very bad hypothesis. And I will show you in a minute how I would grade that. Okay, so introduction includes summary of the background and the hypothesis. Okay, the next section, so in other words, enter, next heading, methods and materials. Don't split that into 2 sections. It's just 1 section. You do not need to list out the materials. The materials are going to be included in your procedure in some method. All right, so this is pretty simple and straightforward. You're just going to summarize the procedure of the lab. I know how tempting it is to just copy and paste the procedure here, but remember, this is going through Turnitin. And if it goes through Turnitin and you include that copy and pasting, you're going to get big time ding for that. And I expect a Turnitin value of 35 or less. That's kind of my goal. Because you're going to have to copy and paste some things. You're going to have to copy and paste, for example, some tables. You're also going to be using the same headings as other people would. So some of it's going to be expected, but anything over 35 is too much. And I do take points off for that. If you copy and paste your entire procedure, I'm probably not going to give you any credit for that at all. All right, so that's category 3 section 3. The next section is the results section. And here is where you're going to put all of your data and calculations. So here's what I want you to do. If a table's provided, use that table and put all of your data and your calculation values in the table. All right. Please do not put your data and calculations in paragraph form. Nobody can follow that, not even me. And I will not give credit for that, or I will take points off for doing that. So put your data in one place where it's really clear what the data is, like in a table. You can create your own tables too if you want to. So I've had students do that often in their lab reports, and that's just fine. As long as your data is in a really organized fashion, and I can follow it, I can see what it is, and I can see everything, then you're good. Now, you also need to include all calculations. All right, so what do I want you to do there? All right, let's say that this first section here is your table, and you filled in all the data that you found in the lab, and you've got some calculations to do that need to be in that table as well. Well, underneath the table, or above the table, whatever you want to do. Some students do it inside the table, and that's okay as long as it's not all scrunched together and I can't follow it. But I think it's best to do it under the table. And what you would want to do is just do all the calculations step by step, line by line, like you would in a math class. As I mentioned, please don't put these in paragraph form. I'm not going to give full credit for that. So, for example, let's say you're trying to find density. So you write the formula for density. Density is mass over change in volume, all right? And then you would say, okay, the mass of this was blah, blah, blah grams. and the change in volume was, I don't know, 15 milliliters minus 10 milliliters. So it's 5 milliliters. And then you would plug those numbers into the density formula and write the density. And don't forget to put your units on your answer. And then you would do that for each calculation, each and every calculation that you're doing. All right, so I will take off an entire point if calculations are missing. And I'm just warning you now. I'll show you where that is on the rubric in a minute. All right. So also one other thing you might have is a graph of some kind. The only lab in 121 that has graphs is lab 8. In 122 there are a number of lab reports that require graphs. So be ready for that. But graphs or figures or anything like that, that should go in the results section. In 122, there's something called pre-lab questions that you need to answer. And those really should be in the results section. I have students that put them all over the place. Don't add a new heading for those. Put them in the results section. And if your pre-lab questions are not there, I do take off a point as well. So make sure you're putting everything in this results section that belongs there. One other thing I want to mention is not all data is numerical. Some lab reports, especially in 122, you might be asked what you observe. Did you observe a color change? Did you observe a precipitate? Did you observe bubbles? What did you observe? Okay, that would be included in the results section. And if there are like questions throughout the procedure that you're supposed to answer, please number those questions and then answer the questions in the results section, numbering them so that I know which questions you're answering. Okay, I don't want to have to look around for the answer for which question is where. I don't want to have to scurry around looking for that. All right, so there's the results section. Then the last section is what we call the discussion. This is basically the conclusion. Please do not call this the conclusion, since GMC Science Department is asking you to put it as discussion. And don't add a conclusion to this in addition. Remember, I only want these 4 written headings here. That's it. All right, so what is the discussion section? The discussion section should be a standalone section that basically, if a reader could read this and know what you did in the lab, were you successful, what were your results, what were the answers to questions, that kind of thing. So first of all, summarize the lab and write down any important values. Okay. Then let's say, for example, you are trying to find the identities of some unknowns using density. So you would want to write down the densities of your unknowns, right? And then you would want to find the identities by comparing those to the known values. So you would explain how you're finding the identities and identify each unknown. Okay, that kind of goes along with the fact, was the hypothesis supported? In that case, yes, it was. If you did that good hypothesis and you found the identities of your unknowns, then that's your hypothesis was supported. And let me just say something here about the hypothesis. If you come to this point and your hypothesis is not supported, you did something wrong. Either your hypothesis was incorrect or your calculations were incorrect, or something in the data set or the results section was incorrect. Something was wrong. So you might want to go back and kind of look at that again, or maybe email me with questions at that point, because something's wrong if your hypothesis is not supported. All right. Also, I want to see were the objectives of the lab supported. Remember, I mentioned to include those in the introduction. So that should be included. What did you learn in this lab? What applications might be in the lab? This last section is good also. I don't take points off for not including this, but kind of discuss what you could have done that would have made the lab easier for you or better in some way. All right. So there's a discussion. Again, as I mentioned at the first with the title page, I would do a page break after this because your reference page should be a separate page. All right. So page break, heading, references, and then list your references in APA format. They must be in APA format for you to get full credit. All right. So that, remember, I expect only these 6 sections. Do not add extra headings or sections, please. And make sure that you are putting your headings on each of the sections as well. That helps both of us out because if you've got a heading there, I know that right here, I should see a summary of the procedure and I should see a summary of the background here, right? So that's really important to put those headings on. All right, so there's what's expected. This is exactly what your lab report should look like. Now, how is this graded? Let's go to the rubric. All right, there are 5 categories in the rubric. The first one is formatting. And even though this is the first category in the rubric, this one is going to be the last one in your lab report. When the lab report is graded, please look through your lab report and read all of my comments, because for each of these categories, I am going to tell you what you earned on these categories and why. And I'm also going to make comments throughout your lab report. So make sure you read all of the feedback, because what I'm trying to do is help you do better next time. All right, so in the first category, this is basically measuring whether you used APA. All right, so you should have a title and a reference page in APA format. You should have the 6 sections, right? The title and reference page are 2 of those, but the 4 written sections in the middle should have headings on them, right? The headings must be there. Don't add extra ones because I also might take some points off for that. It needs to be double spaced, and there should be a minimum number of spelling and grammatical errors. That's kind of obvious, right? So if all of those things are true, you get 4 points. And there's really no excuse for not getting your 4 points on this thing. Students who don't get 4 points are the ones that were not reading my feedback. They're just turning in lab reports each week with the same exact mistakes. They don't ever bother looking at the feedback, and they're suffering for it because they could do so much better if they would look at the feedback. All right, so no excuse for not getting 4 points on this one, right? All right, the second category is introduction and methods. So what I'm doing here is I'm combining introduction and the methods and materials section together. Remember that the introduction section is basically a summary of the background, right? And the methods and materials section is a summary of the procedure. So I'm looking at, did you include everything in those 2 sections that you were supposed to? Let's say that into introduction section, you entirely didn't mention some formulas you were supposed to do and some other things. All right. But your procedure was excellent. So I might give you 3 points for that because your introduction had half the stuff missing, but the procedure was good. So 3 points. Let's say your introduction was horrible. And I have had a few students who are totally off base. It seems like they're not even doing the same lab that I'm looking at, and their introduction is totally off base, but their procedure was excellent. All right. So I'm probably going to give 2 points for that because the introduction basically gets 0, but the procedure was good. So it gets 2 out of the 4 points. So that's how that is graded, that section. Now, remember that the hypothesis is in the introduction section. However, the hypothesis is one of the most important parts of the lab because it sets you up for what the goal of the lab is. If your hypothesis is incorrect, you're not doing the lab correctly, right? So it's important enough that it deserves its own category here, and it does get one. So let me scroll down here. It's kind of on 2 pages here, and come on. It never scrolls correctly. Let's see if I can do it here. There we go. So let's say that you did that good hypothesis, the one I talked about where you compare densities, that would give you 4 points. That's a great hypothesis. Let's see, you did that other one where you said, I predict that unknown A is zinc and unknown B is aluminum. All right, that's a bad hypothesis because you're just guessing. So but you tried, right? You wrote a hypothesis, so you tried. So I'd probably give 2 points for that. Something else I want to mention here is make your hypothesis clear. In other words, write, I hypothesize that, and then write out your hypothesis. That makes it really clear for me. So when I grade, I can see exactly what your hypothesis is, and I can give you those 4 points. Now, if you kind of beat around the bush and you make some statements that kind of indicate that you sort of know what the hypothesis should be, but you don't really state it, I might just give you 3 points for that because I'm not real clear on exactly what you thought the hypothesis was. Now, if you just, you're going all over the place and your one statement kind of makes it clear that you sort of know what the hypothesis is, but not really, I might give you 2 points or even 1 point on that. So state your hypothesis clearly, please. Fourth category is results. I already talked about what should be included in that section. So you kind of know that, so I'm not going to go over that again. And then finally, the discussion is the last category. And that also, I already went over what's expected in that section. So there is the rubric that we use for grading. And remember that I do have that policy that if you email me your lab by Saturday at noon Eastern time, I will do a summary review of it for you and get back to you. Now, if you don't feel like sending me the entire lab report, you just want to know if your hypothesis is on track, send me your hypothesis. I'm happy to look at it. In fact, I'm thrilled to look at it, especially if you're way off base. Because if you're way off base, you're not going to be looking for the right things in that lab, are you? So I would like to help you correct that before you start the lab. So I'm happy to look at hypotheses as well. Okay, is that everything I wanted to talk about here? I think so. Yep, I think so. All right, so everyone, if you have any questions about anything to do with lab reports or, or anything else, please email me and I'm happy to answer. In my introduction in the other video, I did mention that I've been teaching online for 26 years. I started in 1999 before it was fashionable to do online courses. In fact, they were frowned upon back then. And I've been teaching for GMC for over 15 years. So I kind of know what I'm doing when it comes to online learning. So I do understand that getting back to students quickly is important. All right, so that is it about the lab reports. And I'm going to go ahead and stop sharing and end this session.